All right, so we'll get started with the next session. Before we get started, I really want to thank both Federica and Hannah for coordinating an excellent session, and thank you all the speakers for wonderful talks. Really, a lot of great discussion, a lot of great science that is being done in all of these wonderful labs across Florida, and we really thank you for joining us today. So the next session is going to be on the aging brain. As we all know, as we age, there are changes that are happening within our brain in terms of our blood flow, communication between different regions, et cetera. These are all leading to cognitive, neurocognitive changes. And there is greater and greater awareness and need for understanding of what's happening. In my own field, for example, hearing loss has been linked with cognitive decline later in life, and so we're trying to learn as much about this as possible. So I wanted to put together a session where we invited speakers from across Florida, and we have a fantastic 4 talks lined up here. First one is going to be by Dr. Bradlee Heckmann on neuroimmunity, autophagy, and inflammation in the aging brain. He's from University of South Florida, followed by Dr. Jason Richardson on the effects of sex, age, and genotype on neuroinflammation. Dr. Joyce Gomes-Osman from University of Miami is going to follow up with understanding how exercise can be harnessed in terms of neuroplasticity to promote brain health in aging. And finally, we'll close the session with Dr. Thomas Foster from University of Florida, who will talk about the transcriptional markers of brain maintenance and cognitive reserve during aging. So with that, we'll get started, and I'll invite Dr. Heckmann to please lead us off.
Awesome. Well, thank you so much, first of all, Dr. Rajguru, for the introduction and for the invitation. I'm excited to share a little bit about what we're doing here at USF and what the lab's going to do. So obviously, we're talking about neuroinflammation today, primarily in the context of Alzheimer's disease, but we're going to be moving out from that as a lab and exploring these processes in other realms, including infection as well as brain tumors. So as previous, I do have a couple disclosures. I'm listed as a co-inventor on a couple patents, which some of this work is present today. So to really get started, I want to just give you a brief overview of immunity in the central nervous system. And while this is becoming more and more complex as we learn more about the brain and the brain's immune system, we know that the primary immune cell in the brain is the microglial cell, which is a macrophage-like cell. They typically account anywhere from 15 to a quarter of the cell population of the brain. They're different than peripheral macrophages that come from a different lineage, but we know they function similar to resident macrophages in other tissues. And they're an innate immune cell that can self-renew. They can proliferate depending on their stimulus, both pro and anti-inflammatory, and they're restricted to the central nervous system. And what our interest was based on some things that we identified with regard to this autophagy machinery and the autophagy pathway and associated non-canonical functions of autophagy and macrophages in the periphery. We were interested to see what the role of the microglial autophagy machinery would be with respect to neuroinflammation. And I don't have time to give significant background on any of this, and all of this has been published in a paper we had in Cell last year. So feel free to go there for more background. But our real goal was to see what happens with the autophagy proteins in the context of neuroinflammation. And just to give a quick overview of autophagy versus these non-canonical pathways that we call LC3-associated phagocytosis, or LAP, as well as the pathway I'm going to talk about today, LC3-associated endocytosis, or LANDO. We know that there's different components of the autophagy machinery that ultimately function to stimulate the engulfment of intracellular cargoes, leading to degradation in the lysosome. This has been well studied for the past 3 decades or so. But what we also know is that a lot of these components can be shared in these non-canonical pathways, such as LC3-associated endocytosis or phagocytosis. There's also unique components of these pathways, including Rubicon, that delineate them from canonical autophagy and the genetics of canonical autophagy. But there are also shared components. So just keep that in mind. You don't need to know each of them by name for the presentation, but just know that autophagy is shared between these pathways. The regulators are shared, but there's also distinct components, and Rubicon is one that you're going to hear me talk about. So the next question really was, can you see, it's not moving forward. There we go. So the next question really was, do we have a model to study this? So we wanted something that was characterized by neuroinflammation, and we know that autophagy plays a role, especially in neurons, in these processes, but really what's happening with the immune system. So we chose Alzheimer's disease. We know that it's progressive. We know there's no really viable treatment or therapeutic option for it currently, but really it was characterized by this pathology that we can actually read out. So we can measure the progression of disease when we modulate this pathway of autophagy or all of the autophagy machinery and see how it's affecting in real time disease progression in the mouse model. So Alzheimer's was a prime candidate model to study this because we know that it's progressive. We know that it has not only a substrate that can be internalized by microglia, that's beta amyloid, but also that we have inflammation in the brain. And then, of course, we have symptoms. So we can actually measure those in the mouse model. And of course, centric to Alzheimer's and what is thought to really drive disease, at least through the amyloid hypothesis, is this cleavage or aberrant cleavage of amyloid precursor protein into beta amyloid peptides. And those peptides are monomeric when they're cleaved, but they rapidly oligomerize and form amyloid fibrils and ultimately those amyloid plaques that drive human disease that I think most people are familiar with. And also, these amyloid fragments can be internalized, again, by microglia. That's well known. So we asked the question, is autophagy or the autophagy machinery even really playing a role in the internalization of these amyloid peptides? And one protein that we can read out is LC3. So LC3 is a family of proteins, actually, that is conjugated to those autophagosomes or to phagosomes or endosomes, depending on the compartment, subcellular compartment we're looking at, and helps facilitate the trafficking of those vesicles, at least in the broad overview of it. And what we found was if we have microglial cells that are just wild type cells and we administer amyloid in red, you can see that LC3 is recruited to these vesicles. So it suggests that there's some activation of the autophagy pathway. However, if we knock out genes that are required for autophagy, we still have recruitment of that LC3 to those vesicles containing amyloid. Now, if we knock out components of what we identified as the Lando pathway, including a shared component between autophagy and Lando ATG5, we see that we abrogate that recruitment of LC3 to those vesicles. So it's a distinct process from canonical autophagy, yet it relies on certain components of the machinery to function. And again, I don't have time to fully show you all the data supporting that, but just take it at face value there. Also, we noticed that if we abrogate Lando and we administer amyloid to those cells actually become hyper inflammatory. So abrogation of Lando imparts this increase in inflammatory mediators from those microglial cells, including TNF and IL-1 beta, which are known in human disease. So it appears that Lando is really protective against, or this autophagy machinery as a whole is protective against the neuroinflammation. And really, we were able to tease out that that is due to a recycling defect. So typically, Rubicon and all of these components of the autophagy machinery put LC3 on these endosomes that contain amyloid and not only function to degrade that amyloid, but also recycle receptors to the cell surface to recognize subsequent rounds of amyloid that are extracellular. And if we block all of these components, we have an abrogation of this pathway. So we end up basically, in a long story short, end up with extracellular neurotoxic amyloid increasing outside of the microglia and obviously creating this Alzheimer's pathology. And we see that when we have Lando deficiency in a microglia. So we end up with extra amyloid outside, and that drives disease. And again, you can see that since it's published, we think a really convincing story. But the question for us was, how do we link this, essentially making this mouse model of Alzheimer's disease, the 5X FAD model, which overexpresses humanized mutants, worse, to human disease itself. And we had some very nice cohort of brains from Alzheimer's patients with age-matched non-Alzheimer's healthy controls. And what we saw was, of course, they have plaque formation, but what we saw is if we profile the autophagy machinery as a whole, we see a significant downregulation of master transcription factors, including TFEB and EIF5 alpha in these Alzheimer's patients. We also see downregulation of Rubicon, the protein I told you earlier about, gene I told you earlier about, that's involved in Lando, and another gene that regulates Lando, ATG16. And this isn't restricted to the level of the transcript. It's actually at the level of protein as well. So these Alzheimer's patients pretty much across the board have a significant suppression in all of this machinery required for LC3 associated endocytosis. So it got us to this question of, well, what if lando deficiency alone could drive Alzheimer's disease? So trying to relate this to an aging model, if you will, something consistent with what we would see in a human patient. And, we had the opportunity with ATG16, we found that if we delete this WD propeller domain, and I'm not going to go into the biochemistry behind all of this, but if we delete this domain from the protein ATG16L, we actually have a mouse that lacks lando. So they're deficient in lando, but they actually have perfectly fine canonical autophagy. So autophagy processes and everything else are intact, but they are lando deficient. And what we decided to do is take microglia from those animals. These are primary microglia, and I don't have time to explain the assay, but what you're seeing in red is recycled receptor. So this is receptor that has been internalized by the microglia and then trafficked back to the plasma membrane. And if we look at these 3 receptors for amyloid, including TREM2, we see very nice recycling in wild-type microglia. However, if we look at these delta WD or the lando deficient microglia, there's a significant impairment in that recycling. And we've done this in other cell models, not just primary microglia, and it's very consistent. So we failed to recycle receptors. So this is basically confirming that those mice are deficient in lando. And again, that goes hand in hand with earlier work that we did. So really, we asked this question, what happens in mice that are aged, right? So for mimicking human disease, we aged these mice out to 2 years. Control litter mates had no amyloid deposition, really basal level of microglial activation, and then no hyperphosphorylation of this neuronal stabilizing protein tau, which is typically prevalent in Alzheimer's patients. However, if we have lando deficient animals, and they don't carry any associated humanized transgene at all, it's literally just deficiency in lando, we see that these mice have a massive deposition of endogenous beta amyloid in the mouse. And you'll notice they don't form those nice plaques that you would see in a human. And that's simply really most likely due to the difference in biochemistry and biophysics between mouse and human amyloid beta. But they do have this deposition of endogenous amyloid beta. They have very robust activation of microglia in the hippocampus. And then we see hyperphosphorylation of tau, especially in the CA3 field of the hippocampus. And if we move in closer on that, what we actually see, again, we're just reiterating an increase in phospho-tau in the hippocampal neurons, but also what we see is this morphological change of the microglia from the ramified state to this very amoeboid shape, very consistent with hyperactivation towards an inflammatory state. And if we look at inflammation by at least transcript level within the hippocampus, we see exactly that. We see that when we have a lando deficient animal that's 2 years old, they have increased IL-1 beta production, as well as TNF alpha and IL-6, as well as other inflammatory cytokines. So they have neuroinflammation. Not only that, we actually saw that they have very pervasive cell death. So we can measure that by cleaved caspase-3 for apoptosis within the hippocampus. It's very nice staining. And we can actually confirm that by looking at tunnel staining within the hippocampus. So these mice, there's always a potential that cleavage of caspase-3 doesn't result in terminal death. But with the tunnel assay, this is pretty convincing that there is actual neurodegeneration occurring in this model. Consistent with that finding was the fact that we have a pretty significant impairment in long-term potentiation. So the ability to signal across the hippocampus, you can see in lando deficient mice is severely impaired here in black compared to their litter mates. So they have that deficit, and not surprising based on that result is if we measure behavior, whether we're looking at anhedonia and a sucrose preference test, again, consistent with human patients, there's a decrease in their ability to sense pleasure. If we look at more memory and working memory type assays, we see a decrease in the ability to spontaneously alternate in a Y maze. And then, of course, novel object recognition. These lando deficient mice have a severe impairment in their ability to recognize a new object or remember that old object. So there's a severe impairment in their actual behavior. So we thought we created a pretty nice model of spontaneous disease that mimics most symptoms and most pathology of a human patient within an aged animal without overexpressing all of these proteins that are humanized in the mouse. But we asked the question, obviously, the goal of all of this is to get a efficient therapeutic for disease. Can we rescue this AD pathology? And the answer to that was we didn't know at the time, right? So we started with mice that were spontaneously alternating at a lower level, as well as having discrimination impairment with their novel objects. So these mice had established disease at about 20 months is when we started treatment. So they have very established disease as measured by these parameters. We take those animals and we administer MCC950, which is an inflammasome inhibitor that blocks IL-1 beta production. So we're targeting just one of those inflammatory mediators in the brain. And we can see that, not to any surprise, we decrease microglial activation by blocking the inflammation pretty clearly here. So that wasn't too much of a surprise, as this has been reported before with MCC950. However, what was pretty shocking was if we treat with MCC950, again, in mice with established disease, we reverse the tau phenotype. So we actually ameliorate the hyperphosphorylation of tau within the hippocampus. We actually start to restore behavior. So we're able to restore behavior 75% compared to a control age-matched littermate from a mouse that received a control administration of PBS, for example. So we're able to actually restore behavior in this model, which was pretty shocking to us that we had actually such nice restoration of behavior. Also, what I didn't show you, and just in purpose of time, is by treating with this inflammasome inhibitor, we actually reduce active neurodegeneration. So there's still this idea that, obviously neuronal loss is probably contributing here simply because we have, simply because we have not fully restored function. So neurodegeneration probably accounts for that component that's not been restored. But targeting neuroinflammation seems to be a very viable approach to at least alleviate a lot of the pathology in this AD-like mouse model that we've established that's age-associated and spontaneous. And we showed that Lando plays a role in protecting against all of this phenotype. Furthermore, we know that the inhibition of neuroinflammation can rescue the behavior and memory, likely functioning by reducing that tau hyperphosphorylation and then obviously the sickness of those neurons. And then it appears neuroinflammation is upstream of tau phosphorylation and progressive neurodegeneration. And then ultimately, we want to know if we can, go beyond just targeting inflammation and target Lando specifically. And we have a lot of preliminary data suggesting that Lando is playing a very important role in protecting the brain, not just in neurodegeneration, but also in the context of infection, as well as a very strong role in brain tumor immunity. So we think it's a very viable target to pursue. And we have a lot of work, of course, to do on it, but it's something that is very interesting to us. And I think we'll have some hopefully good progress in the coming year or two. So on that, I'm right at time, hopefully. I want to thank, obviously, my postdoc mentor, Doug Green. A lot of this work was done in his lab before I joined USF. Funding from the NCI and the NIAID, collaborators, including Tom Weilman, who was a big help with our age mice. And then, of course, being a newer lab, we are hiring. So if you know of anybody or you yourself are interested in a postdoc position or entry-level staff scientist, please reach out to me. We are wanting to do some amazing high-impact science, and we want the best people. So please feel free to reach out to me, email. And if you have any questions, I'm more than happy to answer after the session or via email. So thank you again to the organizers, Dr. Rajguru. A pleasure.
Great. Thank you, Dr. Heckmann. Wonderful talk, and really looking forward to the discussion at the end. So the next speaker is Dr. Jason Richardson from Florida International University, who will talk about the effects of sex, age, and genotype on neuroinflammation in a humanized mouse model.
Okay, well, thank you very much for the invitation. It's a pleasure to be here and to talk with you today. I'm just sorry we couldn't be in person. Hopefully by next year that will be able to happen. So my name is Jason Richardson. I'm fairly new to the Miami area. I've been here a little bit over 2 years. I'm at Florida International University, the Robert Stipple School of Public Health and Social Work, and where I'm Professor of Environmental Health Sciences and Associate Dean for Research. Just a little quick background. We have a very multidisciplinary lab, multiple research areas. Broadly, most of our research is focused on the interaction of genetics and the environment and the etiology and pathogenesis of neurological diseases and disorders. We have active projects in Parkinson's disease, focusing on neuroinflammation, genetic polymorphisms in transporters. Not going to talk to you about our gene environment in Alzheimer's today, but we do have a very active area in that, and also in attention deficit hyperactivity disorder, focusing on developmental pesticide exposure and epigenetic mechanisms. What I am going to talk to you about today is really our second major area of focus, really for about the past 10 years, on glial cell biology, nuclear receptors, and the development of therapeutic approaches for inflammation. And ultimately, we're very fortunate to have a large number of colleagues and collaborators where we can really pursue our research goals in a multidisciplinary manner. We make use of a variety of research techniques from in vitro. We'll talk a little bit about that today, along with in vivo animal models, but we also do some human population studies as well. So thank you to Dr. Heckmann for a little bit of an introduction. I won't belabor too long on this. Just a few other things to keep in mind about Alzheimer's disease. It is the most common neurodegenerative disease. As we study both Parkinson's and Alzheimer's, it's a pretty large disparity in the numbers of people, and both of those are on the rise. But ultimately, almost 6 million individuals in the United States are suffering from Alzheimer's disease as of 2019. And importantly, there are very large health disparities in this area, much more disproportionately affecting African Americans and Latinx populations. And actually, hopefully some of you will be able to attend. We've got a newly funded NIA conference series to look at those health disparities and gene-environment interactions and Alzheimer's disease between FIU and my colleagues at Emory University that will start next year. But the important part is early onset Alzheimer's and late onset Alzheimer's. Dr. Heckmann talked a little bit about some of the genes that are involved. Early onset are very rare cases. Most of what we're talking about is late onset and sporadic. These occur after about 65 years of age. And of course, we know that individuals experience memory losses and declining cognitive function. And I think it's apropos for the title of the section that we're talking about here is when we start looking at risk factors for Alzheimer's disease, aging is the greatest, and what we're going to talk about is non-modifiable risk factors in Alzheimer's. So more than 96% of the cases are ages 65 years of old and older. This is not normal aging. Alzheimer's disease is not that you get old and over time you're going to get Alzheimer's disease. It's really a hyper form of aging with multiple pathological drivers. And in saying that aging alone is necessary, but it's not sufficient to cause Alzheimer's. And ultimately, the reason behind this remains largely unclear. We're focusing on roles in the inflammatory system. Lipid dyshomeostasis has really been sought as a factor as well. As Dr. Heckmann said, protein degradation and synthesis, be it either through the lysosomal pathway, the proteasomal pathway, or other pathways, is also impacted by aging and also in Alzheimer's. The next part, and this is where the field, at least in the human aspects of things, has really paid fairly good attention to the role of sex and susceptibility to Alzheimer's disease. And unfortunately, in a lot of basic science studies, this is not focused on as much as it likely should be. So out of those 5.8 million cases, around 62% of them are females. Females progress faster in development of Alzheimer's than men. They have higher atrophy rates in the hippocampus and the cortex. Early on, they accumulate more A beta and there's worse cognitive outcomes. As the disease progresses to the late stages, those sex differences start to disappear. But initially, we do see these in females. So the next part is when we start looking at genetics and Alzheimer's disease and how that interacts with age and sex. And there's been multiple genes associated with Alzheimer's. The latest GWAS study was well over 100,000 people. Unfortunately, it was primarily Caucasian of European descent, so it limits some of the generalizability that we can look at. But in those influence genes, the one that has really always been the highest influence that we've known for 30 years is having an ApoE4 allele. And ApoE4 is apolipoprotein E. Let me see if I can manage to get my pointer to work. Okay, yeah, here we are on this. And so ApoE is a multifaceted protein. It's primarily known for its role in lipid metabolism, the role in regulating cholesterol efflux and transfer, but it's also associated with many other aspects of Alzheimer's disease, and particularly the E4 risk allele you see greater deficits in cerebral hypermetabolism, synaptic deficits. You see effects on tau, A beta, and cognitive. But what we're going to talk about more today is this neuroinflammation component. And there has been data demonstrating the effect of ApoE4, but we are typically lacking that as it relates to sex and age. So this is from a review article on Alzheimer's and dementia, and many of you that follow Alzheimer's have looked at this. So there's a number of biomarkers that are looked at to try and get to the point of what is early in the process, what's kind of the prodromal before you start getting AD, and then ultimately what happens at Alzheimer's. And so there's multiple biomarkers, CSF beta, amyloid PET, CSF tau, cognitive task. But what's becoming very important to start to look at is this role of systemic inflammation, obesity, diabetes as risk factors, and whether those early metabolic and inflammation components can drive aspects of neurodegeneration. I think you saw a little bit of that from a different aspect in the previous talk. So we started out with a general hypothesis is that age, sex, and ApoE genotype modify the response to an inflammatory stimulus, and ultimately that female ApoE4 carriers may be more susceptible to inflammation. And so we really sought to determine the genotype and sex-specific effects using LPS, lipopolysaccharide, as a model inflammagen. And to do this, we used ApoE-targeted humanized replacement mice. So these were made by Pat Sullivan years ago. Pat's still at Duke and provided us with these animals. And so essentially, there is the apolipoprotein E humanized isoform that is under the control of the mouse ApoE promoter. These have been back-crossed over 12 generations to a C57 Black 6 Jackson background. And importantly for us, when we're trying to make that translational leap over into the human population, these lack that traditional aggressive genetic mutations that drive early onset AD. Most of the animal models that you see used in the Alzheimer's field do have those rare early onset mutations. And so perhaps these mice may be a bit more relevant in translational studies. There are some reports that they do accumulate A beta and tau. We haven't seen that in our hands, but there's multiple aspects of AD that they do recapitulate in the expected genotype frequency. So how do we go about doing this? So the first thing we did is we isolated primary astrocytes and microglia from postnatal day 0 to 1 mouse pups. We did this in a sex and a genotype specific manner. So what we end up with over here, using magnetic bead separation, are male and female primary microglia, male and female primary astrocytes, and these are either ApoE3 or ApoE4. An important aspect when you're doing any of these in vitro studies is really to make sure that they're exceptionally clean, that you don't have contamination from one cell to the other. These are some pictures. These are primary mouse astrocytes, the E3 and the E4. The green's this is GFAP, and green for the astrocytes. You can see the nice flat shape that we typically see in culture. You see very few, if any, microglia in these. So we get 98 to 99% pure. These are the microglia here. These are stained by IBA1. Again, very pure. Here's an example of a culture that's not as pure, and we don't use cultures that are not 98% pure or more. So here's some initial data that we have in these, and we first start off comparing the traditional mixed sex cultures, which is what you see primarily in the literature. And we use the grease assay median nitrite from the upregulation of inducible nitric oxide synthase as an output measure of activation. So we used a fairly low dose of LPS, 10 nanograms. We used interferon gamma as another challenge, and then LPS plus interferon gamma as our other. And these are initially in primary microglia. So what you'll see here is LPS produces a fairly small response in the E3s. It's much more pronounced in the E4s. When you put the LPS and the interferon gamma, we get a much more robust response. And again, we see the same aspect of E3 versus E4. This has been reported in the literature with mixed sex. From sex-specific primary microglia, this has not been reported in a genotype-specific manner. So what we see here is the E4 males are greater than the E3 males or the E3 females. The E4 males produce greater nitric oxide than the E4 males. And we see this as it goes up with the LPS and the interferon gamma. So even at an early age, this is postnatal days 0 to 1, we're already seeing intrinsic sex and genotype differences in these animals. These experiments were run with charcoal strip media, so we don't have to worry about hormones in the media or anything with that. These are the same assay run in primary mouse astrocytes, and in this, we used a cytokine mix that's got interleukin-1 beta, TNF in it that are reported to push the astrocytes to an A1 phenotype where they're more inflammatory. And again, you see the difference between the E3 and the E4. When we do this in a sex-specific manner, we see even with the E3, the females respond much more robustly. Again, the E4 drives genotype effects, and it drives that even more in the females than it does in the male. We have additional, when we've looked at media cytokine release and some other mRNA measurements, but this was just some initial data to talk about trying to stay on time. So we wanted to move this into an in vivo situation with these animals. We used 2 ages, 3 and 4 month and 16 to 17 month E3, E4 females. We gave a single IP injection of 0.5 milligrams per kilogram LPS. This induces a fairly rapid inflammatory response, but doesn't cause major effects on weight gain or fever. We collected serum. The left hemisphere of the brain was taken and drop fixed after saline perfusion for immunohistochemistry. The right hemisphere was taken for mRNA expression. And initially, we've just done some target profiling. So bear with me a little bit. We're very fortunate to have a really good biostatistics department here, because when you start looking at multiple age, sexes, and genotypes, it can get a little complex. We've got this recorded, so you can go back and take a look at the tables from the multi-parameter ANOVAs here. But these are first serum cytokine measures. We see a robust TNF alpha response, and we typically don't see a ton of effect with this at the genotype with this, but age really does drive a differential effect where we see greater enhancement of serum TNF with age. For interleukin-6, we also see the effect of age, but now we see an effect of sex as well, where both the E3 and the E4 females in the age group reacted much more than the males. These are interleukin-1 beta data and interleukin-10 as an anti-inflammatory. Again, obviously treatment effect, but we also see an age effect here. In the interleukin-10, again, we see an age and a sex effect also, but we do see some genotype effect. When we look at this from a brain standpoint, this was actually pretty interesting to us, and we're going to replicate this. Seeing the young E3 males had a much more pronounced TNF transcript induction response. But we see differences in the frontal cortex and the hippocampus. When we get into the aged animals in the hippocampus, we see greater effects in the E4 female compared to the E4 male. Similarly, with interleukin-1 beta, we have similar effects. Those E4 females that are old show the most robust response following the TNF treatment as well. And interleukin-6, we see fairly similar effects also, although we do see a good bit of genotype driving the effect with greater effects in E4. And again, with the aged animals, the E4 female displays a much more robust response. So where are we at this point? So from the limited data that I've shown you, I think it's pretty clear that ApoE genotype and sex is a significant contributor to the response to LPS or inflammatory stimuli, both in vitro and in vitro. That suggests there's an intrinsic difference between males and females. We also see some regional differences between the cortex and the hippocampus. And ultimately, I think overall, it's important, and these data support the idea that genotype, sex, and age are critical factors to consider as it relates to inflammation in AD. So in the future, we're in the process of performing RNA-seq and shotgun proteomics on these samples to better understand the effects of genotype, age, and sex on the response to inflammatory stimuli. I do want to acknowledge the people involved, an outstanding postdoc who's now going on to an independent position, a CLE, Isha, as a finishing graduate student. This is really her baby, as well as Oren Bursak, our chair of biostatistics here at FIU, and Yoon Hee, who is my lab manager. This was supported by NIHS through my R01 and then through a collaborative supplement with Duke University, where we're looking at some similar things in a population-based study as well. So thank you very much. I hope I kept us on time, or at least fairly close to on time.
Absolutely. Thank you, Dr. Richardson. And yes, we hope that, by next year in the 5th Neural Engineering Symposium, we'll be in person. So we'll see.
Great, thank you.
All right. So the next speaker that we have is Dr. Joyce Gomes-Osman from University of Miami. She's going to talk about exercise, harnessing neuroplasticity to promote brain health in aging.
Thank you so much to Dr. Rajguru for this invitation. I'm really excited to talk to you today, but I have a big disclaimer. I'm not an engineer. I'm a rehabilitation neuroscientist. And so the stuff that you're going to hear from me is going to be very distinct from the wonderful presentations that we did. But I'm going to be presenting to you about the idea of assessing neuroplasticity in humans and in translational studies that are using exercise as a means to improve cognitive health in people who are aging. I'm an assistant professor for the departments of physical therapy and neurology here at University of Miami. And so for this group, I really don't have to tell you much about the importance of maintaining and promoting cognitive brain health. We've heard a lot in the previous sessions today, but another side of it, a slightly different side of it, is that when you ask people who are aging, what are the biggest health-related priorities, they overwhelmingly will tell you that maintaining a sharp mind is something that's really important to them. And I have another on the right side here, just a paper that, you don't have to scroll through there, but just to highlight that despite all of the fantastic advances that we've done in order to understand the processes, to understand the mechanisms of age-related cognitive decline, we really don't have wonderful therapeutic targets right now. And so the success rate is actually 0.4% or 99.6 of a failure rate, if you want to look at it like that. And so this really begs the question of right now, if we don't have anything right now at this moment for people, what are other things that we can be doing? And so one of the things that still strikes me as so interesting is the fact that 35% of the risk of developing dementia is caused by modifiable risk factors. And so this is really interesting study published in Lancet a few years ago that was actually trying to estimate, and so they used population attributable fraction, which essentially calculates the percent reduction in new cases if a specific risk factor was to be eliminated, right? And so this is how they got the 35% of modifiable risk factors for dementia. But it's really striking to me that among all the things, right? Smoking, keep learning, check your hearing, exercise by itself only confers about 3% reduction in future risk if everybody now were to be active. But if you group exercise, right, if you group things that coexist with exercise, such as being social, such as managing blood pressure and blood sugar and maintaining healthy weight and managing depression, this grows to about half of that 35%. And so exercise is the most effective therapeutic we currently have to mitigate age-related cognitive decline. But there's a big challenge with exercise, which is it is very variable among individuals. And so some people have excellent an excellent response when they start exercising and they see that the cognitive benefits are really, really valuable to them, but others really don't. And so we are really, in my lab, we're really trying to understand what is the source of this variability. And so our long-term goal, right, is really to develop individualized exercise interventions to promote cognitive brain health and aging, because this big variability in the cognitive response to exercise really tells us what we've learned, not just from cognitive aging, but what we've learned from cancer, for example, that a one recipe for everyone really doesn't work, that these that successful interventions really need to be individualized in order to have the greatest gains. And so in order to look at that, right, so we know that exercise is effective, right? That has been demonstrated greatly. There are about 50 meta-analyses that together have found the same result, which is exercise is effective to maintain and improve cognitive health in people who are aging. And so why is it so variable? Well, that's a really, really complex question with a complex answer. And so when you look here, the nervous system rearranges itself in many, many different ways following exercise. We've heard a lot about neuroinflammation, which is one of the mechanisms as well. But exercise decreases hippocampal atrophy and improves neurogenesis, decreases atrophy and white matter tracks, and improves the metabolism of the brain and blood flow of the brain, and has all these effects with neurotrophic factors. And so, but when we go back from the human looking at great variability, and we go back to the extensive literature that exists in non-clinical animal models, we really see that synaptic neuroplasticity is the mechanism that sort of ties all of those mechanisms together. And so we really look at this as the way that the functional implication of improved hippocampal neurogenesis, along with increased brain-derived neurotrophin factor and vascular endothelial factor. And so all of those mechanisms work in concert in synaptic plasticity. And one plasticity has many, many forms as well, but neuroplasticity in the form of long-term potentiation is one form of neuroplasticity that has been shown to be critical and necessary for exercise-mediated cognitive improvements in animal models. And so one really exciting thing that we have in the lab is that we use non-invasive brain stimulation, which allows us to gain insights into neuroplasticity, into long-term potentiation like neuroplasticity. And so this is essentially done with transcranial magnetic stimulation. And so modeling the same paradigms that were used for LTP experience in animal models with non-invasive brain stimulation, we're able to get this neuroplasticity response, right? And so essentially you assess the excitability of the whole system. And so we look at the brain as a complex system. And so by assessing the motor evoked potentials with surface electrodes applied to the hands and stimulating the brain with TMS, the first thing that we get is a baseline response of excitability. And after that, when we give the same plasticity inducing stimulation that are akin to the one that are used in animal models, we see this potentiation of the response. The local response is really measured by these MEPs that grow, right? And so you can see here the potentiation, which is transient and lasts about 30, 40 minutes after you give the stimulation and returns to baseline. And so here, what I want you to pay attention is that and those first minutes, 5-10 minutes, are really the most robust and reliable intervals where you can see this local response, local neuroplasticity response. And so, again, we're looking at the brain as a complex system here. We measure the local response to this perturbation that you can think of with motor evoked potentials, but you can also take a look at a more global response by just combining EEG with your measurement. And so you can look at several different outcomes of EEG, but one that we've started to look at is the global mean field potential, which is sort of a combined global assessment of excitability that's not just constrained to the motor cortex, which is which is what TMS, we know so much about TMS, but it's sort of, we really need to go beyond the motor cortex to those non-motor networks that are so critical for cognition and for understanding exercise-induced cognitive improvements. And so here is a little bit of the theoretical framework that we talk about, right? And so neuroplasticity, specific neuroplasticity in the form of LTP, is something that declines with age. And it coexists with a lot of things that are important, right? Some things that allow us to look at the mechanistic aspects, such as decrease in brain-derived neurotrophin factor, but importantly, cognition and quality of life and an increased onset of mild cognitive impairment and dementia. And so an interesting thing that we're capitalizing on is that overwhelmingly in animal models and in humans, exercise improves cognitive function, and is really thought to be critically mediated by neuroplasticity, which coexists with many other things, right? The trophic factors, cerebral perfusion, and to improve and promote cognitive brain health. And so we have, I have a KL2 from the Clinical and Translational Science Institute here at University of Miami. And an interesting story about that when we talk about how COVID has impacted this. But we set off to look at a simple study, to do 8 weeks of exercise in people who are aging, so at least 55 years of age, and who don't yet have cognitive impairment. And so some of the slides that were shown about synaptic changes that precede many in decades, right? The actual onset of cognitive symptoms. And so we wanted to come a little bit earlier in the lifespan with the idea that, sometimes, when you're earlier on in healthy aging, something can be done about it. And so this is just aerobic exercise, 4 weeks of moderate, 4 weeks of high intensity, not because we think that aerobic exercise is better. We could have a whole other talk about that, but because it's the most easy way to quantify the physiological adaptations that are going to occur as a response to exercise, so that we can really pinpoint what are the active ingredients and get to the stage of individualization. And so we have neurocognitive assessments. We have this neuroplasticity response that I talked to you about. We're also looking at APOE allelic status and brain-derived neurotrophin factor, because that is one of the trophic factors that are most consistently improved with exercise and correlated with cognitive outcomes. There's also a SNP in the brain-derived neurotrophin factor gene that essentially impairs the activity-dependent release of BDNF, and therefore would also render some people plasticity impaired in a way. And so that's an interesting angle that we're gonna look at as well. And so before and after, so this is just a feasibility study, everybody gets exercise and we see what happens. And so in terms of preliminary results that I have to discuss with you today, some exciting things are happening. And so we're looking at here at the repeatable neuropsychological test battery on a global cognition assessment. People do improve significantly from pre to post. But the interesting here, the thing that I wanted to show you here is that, yes, well, the mean, Bolden, right? Everybody responds on average, and the majority of people are responding in the dashed lines here. There are some people who don't respond. And what we want to understand is the variability in this response. And so an interesting thing that's happening as well is that we have, I think, there are about 25 individuals who've completed the pre-post test at this point. And the preliminary data also suggests that plasticity is improving from pre to post. The interesting thing that happens is that, as I showed you, right, compared to the baseline response, we really expect to see a potentiation of the response in these 5 minutes, 10 minutes, 20 minutes, right after the plasticity inducing stimulation or the intermittent theta burst stimulation is given. But what we actually are finding here, which is super interesting, is that we seem to be bringing people back to a normal state from a sort of a hypoactive state. And so we do essentially recruit people who are at least 55 years of age and don't have contraindications to exercise, but they have some, if they have controlled diabetes, they can come. If they have controlled hypertension, they can come. And so it's really interesting to think of the idea that people in this, that at baseline, people are sort of hypoactive in terms of their plasticity, and to investigate the behavioral relevance of that, really. And so another way for me to show you, when we zero in into the intervals, the time points where the literature really tells us that there are these more consistent potentiation of responses that we expect to show this neuroplasticity index, you can see more clearly here that really at pre-exercise as a group, they're sort of hypoactive. They show a response that we don't expect. And this is sort of reestablished to what we should see in a baseline in somebody who's really, really young and otherwise healthy. And the other interesting thing that we've seen, and we just got this data last week, and so I wasn't able to get a lot of interesting ways to show you and tease it apart here, but the brain-derived neurotrophin factors, just serum levels, are also going in that same direction. An interesting thing that's happening as well with exciting about these results is that some of this variability in neuroplasticity is explained by clinical measures of cardiovascular health. And we know that cardiovascular health is linked to brain health, right? That's one of the main mechanisms by which exercise can actually do anything to improve cognitive brain health. But nobody has really shown that a measure of, long-term potentiation like neuroplasticity in humans is correlated sort of in a robust measure with things like, resting systolic blood pressure and diastolic blood pressure and the rate pressure product. And so these are interesting things that we're gonna be able to tease apart in the next few months. Another interesting thing that we are looking at is that, so this is not your just run-of-the-mill exercise and cognition program. We actually have people with their polars, and we're quantifying how much they adhere to this target exercise that we're giving. And we, it's a wonderful team that I work with very motivational graduate students and staff are really trying to get people to get in the target and stay there. And also we are measuring their perceived effort, because obviously some people take, calcium channel blockers and things that alter your cardiovascular response to exercise. But one of the interesting things is that, even though we're with them in the gym, this is all pre-COVID, by the way, we're with them at the gym, we're telling you, bring your heart rate to this level and let's stay there. Every 5 minutes I'm checking. And how do you perceive, how hard are you working, from 6 to 20 or whatnot. People have a great variation in their cardiovascular adherence to exercise. And so it's really interesting for us to think, could it be that this person here, and this person here are going to be the ones that have the least amount of gains of exercise for their cognition in this study? And so obviously we're gonna take a look at that as well. And so then COVID happens, right? And so I think for all of us, whether we're doing studies with cells, with animals or humans, the COVID pandemic has been challenging. But we really have this concern that when normal life returns, because we're interested in capturing data and engaging with older adults and bring them to do exercise studies. So in these studies, people are actually coming to downtown, to the university, to exercise with us 3 times a week for an hour for a total of 8 weeks. And so these people now are going to be considered high risk, right, to leave their homes, a lot of them. And so we needed to pivot, right? We needed to do something. And so we started to think, could we develop the processes to carry out the same study in a remote manner? And so this would require a remote neuropsychological test battery. This would require a remote brain physiology assessment. And so there's an interesting assessment that we're piloting, but also physical fitness assessments, and deliver the exercise fully remotely. And I'm excited to tell you that we're starting and that we're going strong, because so the TMS is what we use as a perturbation measure, right, to get this potentiation of MEPs, which is our measure of neuroplasticity alongside with EEG. But we're working with a company that actually has a brain stimulation device with transcranial alternating current stimulation, but also associated with EEG, because then we can use the alternating stimulation to deliver a perturbation akin to what we do with TMS, and assess the result of this perturbation remotely in the person's house. Just send this the device and carry the assessment, just like I'm talking to you here. And so this is also in the pipeline. And so the way that we actually currently have 16 people who are engaged in the study and set to finish this 8-week exercise intervention before the end of the year. And so what we do is that we ship a kit, a study kit containing, you know, a pulse oximeter, the brain stimulation device, and a sphygmomanometer, and things, you know, a polar, things that we're going to need to assess them. And we are doing all of these assessments remotely. We haven't started with the brain stimulation assessment yet, but we wanted to pilot all of the other data before, which is really important because let me tell you, even though we can talk here, and there's, you know, 60 of us to exercise, we really need to limit the room. And so there's some technological considerations that are really important for exercise delivery that we're trying to figure out and figure out really well. And so that's it. We do the 8 weeks exercise remotely. We do the neuropsych remotely. We do physical fitness assessments remotely. Questionnaires and everything, and then they ship the kit back to us. And so this is a little bit of the video, the home program that we've delivered. And so these are my lab staff, and so I don't have videos of our subjects yet, but it's sort of the same thing. I just wanted to show you how it goes. And so we have these, you know, just a standard sort of on Zoom exercise, but also the non-standard, you know, very careful characterization of how their heart rate changes throughout that program and every single day throughout the course of the weeks, and how their perceived effort changes. And so these are the ingredients of us trying to figure out how, what is the physiological adaptation that this is going to have in their cardiovascular system to then relate that to neurocognitive measures into brain physiology. And that is all that I wanted to share with you. I hope that I've stayed within the time, and thank you again.
Absolutely. Thank you, Dr. Gomes-Osman. Really fantastic talk again, really interesting and exciting work. And I just wanted to say that everybody is a neural engineer, so you can go ahead and please use that term. All right, so the last talk that we have is by Dr. Thomas Foster from University of Florida. And he's going to talk about transcriptional markers of brain maintenance and cognitive reserve during aging. Dr. Foster.
Right. I've been listening to all these talks, great talks, and I keep thinking about why we're here. And one of the reasons, of course, is using artificial intelligence and deep learning to answer questions that we've brought up. So I'm going to kind of summarize some of this and ask the engineers to kind of think about this, and of course, think about it more with my talk. But the output here that we're all interested in seems to be cognitive variability, right? Those that are unimpaired and those that are impaired. And we think it's due to certain variables, pathways in this case, biological pathways such as inflammation or synaptic plasticity. But there's also relevant variables. There's sex, there's genetics, there's independent variables that we could modify. There's diet, there's exercise. So it's so broad and so, it's going to take a lot. It's going to take artificial intelligence to actually get to the bottom of this. What I look at is I look at molecular changes in the brain that sort of relate to all this. And I'm really interested in how we either maintain the brain or something called cognitive reserve. And this has been brought up initially by human studies. And the idea is that when the brain sees aging stressors, whether it's inflammation, oxidative stress, or pathology such as A beta, it has the option, let's say, to undergo plasticity in order to handle these stressors and maintain cognitive function. So the study of cognitive reserve requires these measures of variability in cognitive function for specific circuits within the brain. And some of the circuits I study would be hippocampal dependent episodic memory, prefrontal cortex, and attention. And then from that, we need defined senescent markers within these cognitive networks. And that could be anything from MRI structural changes of the brain size or the region size, activity within that region, or, as I study, gene expression. And then finally, we need the ability to differentiate cognitive reserve mechanisms, so to operationally define these mechanisms. So I have the two main mechanisms up here, and that would be brain maintenance and cognitive reserve. So I'm going to go over these before I talk about the genes, or as I talk about some of the genes. So brain maintenance relates to factors, and that could be genetics, early life intervention, or differential experiences that delay or prevent both cognitive decline and brain changes associated with aging or pathology. So perfect maintenance, an 85-year-old who has a perfect cognition, does not lose cognition, has no change in brain size, activity, or genes. It doesn't happen much, but the idea is this is something that's ongoing before the aging stressors actually appear. Once they appear, you can get other changes that maintain cognitive function. That's cognitive reserve. Again, it's these plasticity or compensatory processes, in many cases, triggered by aging or pathology. So let me show you how that works with genes. If we look at, let's see if I can get my cursor here. Oh, there it is. Okay, if we look, back up. If we look at the expression of all the genes for the young animals, and then we separate our aged animals, and I do work with rats, aged unimpaired and aged impaired. The aged impaired animals that are showing either decreased expression of, say, synaptic plasticity genes or increased expression of inflammation, they're unable to maintain the brain function. And so that's part of the reason why we're seeing an impairment. In contrast with cellular resilience or cognitive reserve, what we see again is the expression of the young animals, and now we see large changes only in the aged unimpaired animals. And so this is a response to aging and or pathology, and it enables the maintenance of brain function. Okay, so I'm going to give you an example of brain maintenance from a recent study we published, and I'm very interested in the changes that occur in middle age. And so some of the changes that occur in middle age in rats and humans would be loss of episodic memory and the ability to pattern separate. That is to recognize and remember stimuli that start to overlap in their similarity. And so for this task, we use a water maze. It's a pool of water. And in this pool of water, sorry about this. In this pool of water, we have two platforms. One has two stimuli. One has a platform that's an escape platform. And so we can make them different distances from each other. If they're far apart, it's very easy. But once we start to bring them in close together, in this case, 45 centimeters apart, particularly when we release the animals from the equidistant position here, they have to make that choice. And we can see that our aged animals are having many more errors relative to the young, although some of the aged animals, and these are middle-aged animals, perform just as well as young. So then what we've done here is to do a principal component analysis on gene expression within different regions of the hippocampus. So these are some of the components. We're looking at the dentate gyrus, CA1 region, and CA3 region. Now we know this task depends upon an intact dentate gyrus, so we expect changes there. But the thing to notice here, and I think that the way AI could work is to differentiate these out. There's changes that occur by region. If I bring in now age, the small plus signs, those are the young animals, which you can see are different from the aged animals in almost every case. And then now if I bring in cognitive function, the aged impaired animals, and that's the circles here, they start to differentiate much more from the young animals, and the aged unimpaired animals look more like the young animals. So it turns out this is a case of maintenance. Animals that can maintain these genes also are maintaining cognitive function. So then the question is, what are these genes? What pathways? Synaptic plasticity, neurogenesis. So we've done this throughout the brain, different brain regions, different tasks, and we find that certain pathways are common, not the same genes. For instance, synaptic genes decline with age, but not the same synaptic genes because each brain region has its own transmitters, its own inputs, etc. Inflammation is always increasing with age. So this is from our original study here, showing across age, and the green is inflammatory genes increasing across age, was not a good predictor of cognitive decline. Here's in the prefrontal cortex, and age, and so the red means increased expression of the immune response. But when we start to dive deeper into this, what we can see is that the cognitive changes do correlate with some of these genes. So for example, For impaired animals, when we see an increase in inflammation genes, these are the immune response genes, and many of these were talked about before, the cytokines, the nitric oxide, the oxidative stress genes. In contrast, aged animals that maintain their cognitive function are turning on genes, again, increasing expression of immune regulatory genes, the ability to shut down or to resolve the inflammation response. The last two concepts I want to talk about are network compensation and network efficiency. Again, this is a type of cognitive reserve. With network compensation, then, we're now starting to look across brain regions. I think that's also going to be very important for artificial intelligence and this deep learning. So when a different, in this case, compensation is when different brain regions are recruited to compensate for impairment of the circuit that's normally involved here. So this can involve a strategy shift. For instance, the hippocampus is involved in space, and if you can't process that space, you might start relying on a cue response, which depends upon the striatum. So you're going to see differences in activity in these regions. Another way this could occur is by actually dedicating a circuit to the process, making it more efficient. So we call this network efficiency. So in this case, plasticity permits less activation in order to produce the same or better performance. And we found this in the medial prefrontal cortex for animals that were trying to find out if they maintain the same behavior, if they can shift. So an older individual you may have heard may be the curmudgeon who doesn't want to change at all. And that may be a problem for some behaviors. What we find in these animals is, in this case, AU means aged unimpaired. For activity-related genes, that would be ARC, FOS, early growth response genes. These are the immediate early genes that tell you that these areas were active. The aged unimpaired animals are showing decreased activity in this area. Maybe it's another area that's taking over, but we think that really what's happening here is they've dedicated part of the prefrontal cortex to this task. Now, it may be that they can't have, they have poor vigilance. So another prefrontal task may differentiate these animals in another way. But in this case, we think that they're having a plasticity process to increase efficacy. And that was by Lada Ionov, who's also a graduate student coming out of my lab. So in summary, measures that what we're going to need here in terms of artificial intelligence and deep brain learning are measures of variability in cognitive function, the ability to detect the cognitive strategies that are being employed. And so this is going to take lots of behavioral analysis. We're going to need defined markers, in my case, gene markers across cognitive related networks that change with age and are associated with variability in cognition. And then we're going to need the ability to differentiate the effects of relevant variables, that would be age, cognitive function, sex, differences in transcription platforms being employed, and independent variables that we think tap into cognitive reserve. And that would be exercise, diet, the history of experience, whether you're social or not. This would be environmental enrichment or even possibly previous inflammation that you've had. And so I need to thank the people who really did this work, the graduate students. I've showed you Garrett Smith's and Lada Ionov's work. I need to thank the National Institute of Aging. I've been funded since 1991 by them. I'm the McKnight Chair down at the University of Florida, so the McKnight Brain Research Foundation, and then thanks to my hippocampus. So I'll turn it over to the moderators.
Great. Thank you, Dr. Foster. So it looks like there's still hope for us to be the super cognitive agers yet. We may just have to exercise more. We may have to try and reduce our inflammation and continue to thinking about our behavior a little bit more. So thank you for, again, like, wonderful 4 talks. I really appreciate all of you. And so now we can open it up for any question and answers that we have, including from other panelists. If you're a panelist, you can unmute yourself and go ahead and ask. Otherwise, you can post your question in the question and answer area.
Okay, I'll give it a start. So Joyce, I liked your talk and the idea that you have to go back and see the baseline response and their initial response to try and determine whether this is correlated with cognitive function. Of course, I want to get back into previous history, even before that, were they active as kids? Have they had inflammation? And then let's sequence their genes. So.
Yeah, I think that that what you bring up is really, really important, and I'm particularly excited about your excitement about talking about middle age. Another work that I didn't present today, but there'll be a paper coming out, is that we actually did some structural analysis and looking and trying to relate cognition and cardiovascular health in middle age, but like earlier in middle age. And there's a lot of really interesting patterns emerging. But I do think that one's history of life, right, of exercise, of movement, is so important. And it would be really interesting for us to be able to talk about that. But it's surprising to me that even with the slides that we have, the knowledge that we have, that these there are some structural brain changes that are happening, and I'm 37, and so it's definitely here. And but the brain health conversation really happens much later in life. And so I think that from what you're talking about, it's really interesting for us to think about, let's create a brain health plan right now, because it's when these things we can steer, right? There's some level that we can't steer, and so there's 35% of the risk that we can steer.
So, but related to that are, again, some of the predictive variables, whether you have E4 or TREM2, and that may even determine what your course is and what your treatment is.
I completely agree with you. And we haven't looked at our APOE yet. And this is a small sample, this is a feasibility study, but we will, and in the future that we'll create, but maybe even put something together. But one of the interesting things that has come out in the literature is that there's this paradoxical effect of APOE carriers responding better to exercise, but the mechanisms are not really well understood. And so I do think that it's not like the non-responders there's nothing we can do for them, but maybe instead of 8 weeks, they're going to get 16 weeks, right? And it's going to be, we're going to have to individualize thinking about that. And to be honest, you're right. Maybe from the work that we have, I can tell you that even if you start as, in your 70s, you can have improvements, but you're right. We probably have to start in childhood.
Do we want to start taking anti-inflammatories when we're in our mid-age?
That's a great question, and that's something that I think we can pose to Dr. Richardson and Heckmann, maybe.
Yeah.
I think it's something that from our perspective that we kind of want to pursue, right? So taking NSAIDs that would typically, hopefully not have a cardiac side effect, something of that nature. If we could, administer those in mid-age and on, if that has any viable effect in mitigating any of this, right? And there's been some studies that have shown I think it was malphonic acid, I think it was. That showed a little bit of benefit. So the idea would be is it enough? Does it offset any potential side effect of the compound, whether you're trying like COX inhibitor or something like that, to the cardiac health to be able to have an effect in the brain? So that's a study that we've thought about doing, actually, is in these mouse models, just putting them immediately on as soon as they wean, putting them on NSAIDs to see if there's any alteration long term. So we'll see.
But the other,
I think that's a good point. I think we've got better data in Parkinson's disease on that currently, and certainly what I like about it now is starting to move the conversation from treatment to prevention. As we all know, by the time, whether it's Parkinson's or Alzheimer's, by the time you show up impaired enough to be in the clinic, there's so much damage done that things that might actually be neuroprotective aren't going to work in that instance. Now, there's multiple reasons things don't translate from the lab, but I think that's definitely one of them. I think the other point, I think all of you kind of touched on this when we start thinking about this idea, and particularly Dr. Foster, about aging and the inflammatory environment. And I think this speaks to the exercise as well. We've always kind of thought it like the teeter-totter. You start off, you're younger, you can handle oxidative stress, inflammation, and then you get to a point where it just kind of tips over. But I think if you follow some of the aging literature and really some of the traditional psychology literature looking at exercise, I think what we're starting to see now is it may not just be that tipping point. It may be that as you age, even though it's a pro-inflammatory environment, you're not able to resolve that inflammation as well. And I think that's something that is an emerging area. There's a little bit of data out there on it, not particularly in the appropriate disease models. But I think that may be another way for us to look at kind of modulating this rather than the broad aspect of knocking down inflammation, because we all know inflammation can be important in certain ways. But can we target that resolution process? Because we see that in Parkinson's models, and we've shown that particularly with an LPS model, is you progressively lose neurons and then you resolve it and it stops. But if you've got older animals that don't resolve, things keep going on with that. So I think it's just something that, in exercise, old animals, same thing, they don't resolve as well as a younger animal that you're running with that. So I think it's a lot of things, interaction, and certainly sex and genotype play a difference, as was alluded to as well.
I think that the other thing that we're going to have to consider is not only brain inflammation, of course, but systemic. But I'm now thinking about the antioxidant literature, and it was thought you take antioxidants, and now we know that's not the thing you do, because your body, your brain is plastic. You want to have a short burst of stress, maybe, and certainly in your muscles, a short burst of inflammation when you work out, and that helps turn on the system for resolving all these. It turns on the antioxidants internally, and then the stressor's over, and they go, well, let's just get rid of the detritus that's here from aging.
I think that's a very, very valid point. And especially, we even see that in our Orlando models, right? The idea is with, this concept of inflammation can be a very beneficial thing, right? And especially if it's in, and even take it into the realm of infection, right? And, it's a protective process. And so I think it's a very viable point in the fact that it's all things kind of in moderation, right? So we want to be able to have those reactions to occur. But at the same time, it's when it becomes that unchecked, out of control situation or progressive chronic situation that you start to have damage peripherally, both, as global inflammation or within the CNS as a whole. So I think it's a fine line that needs to be fine tuned, right? And, part of the thing is, if you look at, a younger organism, it works pretty well. And it's just when all of these processes start to get a little bit out of kilter that they compound. And I think that's one thing that, trying to go back to, and I know Beth Stevens works on this quite a bit with microglia, going back to that idea of the developmental aspect of biology, and trying to be able to replicate that, as you age. So it's something that's definitely interesting to pursue.
Yeah, I think that the last thing that I would say is that one thing that's really hard for us, and I think that probably is it's not just something that happens with us, but exercise is part of a lifestyle, right? And so I'm sure that if you were to look at what exercise does to BDNF, and you'd create a pill and you gave it to someone, you'd get something. But the thing is that exercise helps with your insulin regulation, and that by itself has its own independent relationship with cognition. And then, the cardiorespiratory fitness is another thing. And somehow the body is sometimes, right, pretty smart and understanding that exogenous, anti-inflammatories are different from endogenous and anti-inflammatories, such as things like exercise. And so it's going to be really hard to individualize things because of that. But also we should really think about it's so hard to get deep enough in your area and not consider the other ones, but we really need to do that in order to actually answer the question. And so that's the conundrum that sometimes I find myself in these grant applications and stuff.
Yeah, Joy, I mean, you really hit the nail on the head with that, because it is multifactorial. I like that you present the data, that's been published on the population attributable fractions. And if we look at controlling insulin, and we look at controlling blood pressure, you're talking about a significant drop in risk from those two modifiable factors. The problem I think that we all see just from healthcare component is, it's multifactorial. Number one, human beings as a kind of a broad thing don't like to be disciplined enough to do those long-term things that they need. The other part that we see, and again, kind of giving the plug for our conference and what we're trying to do is, that doesn't apply across all races, ethnicities, socioeconomic conditions, those type of things. I worked with some colleagues out of Texas, large Mexican American cohorts, and what you see in cognitive impairment and AD in that population is it is very metabolic syndrome driven. You see a different panel of biomarkers, you see different things. Some of that is genetic, and we've got great people here, including at with Peggy Perkak-Vance, looking at, okay, where do you inherit your risk alleles from? Because let's take, for example, in a Mexican American population, you're going to have indigenous, but you're also going to have Spanish, and they've shown in multiple other populations, depending upon which lineage you got your ApoE allele from, it can confer either greater effects or lesser effects with that. So I think why I really appreciate being involved in this session is, this is what I love and what I think we need to do, which is that multidisciplinary nature of things, bringing those people that think about it in a different way to attacking this big problem. Because I'm the oddball here. I'm trained as a toxicologist. I wandered into neurodegenerative many years ago, and Alzheimer's even more recently. But I've seen so much stovepiping with things. You're either in the beta camp, you're in the tau camp, now you're in the inflammation camp. And it's like, no, they're all part of the situation. We have to look at it in a more holistic fashion, and nobody can do that on their own.
Absolutely. Dalton, did you have another question you wanted to ask, or Dalton?
Yes, I was just got out of study section, and we're talking about the lymphatic system, and I didn't hear that mentioned today. And I was just wondering, I think maybe Dr. Heckmann, in terms of autophagy and how that could actually be related to the drainage of toxins from the brain. And one more thing, we didn't mention sleep today either. And that seems to be a huge area of research now in terms of normal aging, healthy brain. So I'll just throw that out to the group. Fantastic presentations. I'm glad I heard inflammasome mentioned as well along the way. That's the area that we're studying, but great. Thank you all for coming. The lymphatic system, what do you think about that?
So yeah, so I think it's a very obviously a hot area, and I definitely think it's playing a role, especially in our models. And of course, so one of the other projects that's interesting, you bring up sleep. So that's something that we're going to be pursuing, is this idea of sleep fragmentation, right? So not necessarily deprivation, and not even changing circadian cycling, but just mild sleep fragmentation over the course of a lifetime, right? That imparts these aspects of bouts of inflammation over a period of time. If you combine that with infection biology, right? So having, defense against infections over time, are those contributors overall to disease establishment and altering this whole neuroimmune architecture. The idea that lymphatics, I think that's a really pertinent area. That's something that we've been collaborating a bit with. Just actually got off the phone with them earlier, John Kipnis at WashU, now that he's moved, to try and really profile. It's something we're not set up to do here yet, but really profile, how we're clearing substrates from the brain, right? So the one thing with this Lando pathway that we just don't know yet, why is it altering neuroinflammation, right? Is it truly just an alteration of where we're not clearing? We have a constitutive signal, let's say, within those microglia that just keeps this loop of inflammatory activation occurring, or are there actually changes in lymphatic flow, blood-brain barrier permeability in these animals, where we're actually not necessarily clearing cytokines from the brain, right? Because if we use a sleep model, we have a sleep fragmentation model where we actually administer LPS, and we see that LPS is getting into the central nervous system, is actually crossing the blood-brain barrier, getting into the brain, activating these microglia. And with fragmentation, we actually can induce death in those animals, mortality in those mice, with what would typically be a sublethal dose of LPS. And we see this perpetuation of signaling. Now, the question there is we're not far enough along yet. Are we preventing the clearance? Are we having just hyperactivation of microglia? Or is there obviously a component of systemic inflammation that, you know, is playing a role with cytokines coming from the periphery? We have a couple models that look at microglial specific deletion of TLR4, really suggesting against this idea of the peripheral cytokine pool being the major factor. But it's definitely all very interesting. And I think it goes hand in hand with the idea that it's not limited to one factor. I think there's going to be so many different components that all come together to really play a role here. But it's definitely interesting to bring those up because I think that's definitely where the field is going, especially with the glymphatics, and being able to now monitor glymphatics in a much more in vivo environment is going to be prove very beneficial, I think.
Great. All right. Thank you so very much again, all four of you. And I probably couldn't have closed this session on better sort of a description than what Dr. Richardson said. Is like, you know, we really need the whole reason while we were trying to do this symposium, and we've been trying to do this every year, and we have grown from having 60 or so to about 200 registrants this year in 4 years. And these are all just from universities in Florida, is to essentially promote that, is to promote not only, exchange of information and science, but also to try and spur collaborations across our institutions, bring up these multidisciplinary teams so we can tackle some of these bigger challenges and bigger problems. So we would love to continue having these discussions with all of you. We would love to continue making sure that our universities are staying engaged. And with that, what we'll do is we'll transition into the last session for the symposium, and that is the poster session. So very quickly, let me show you. So what we have done this year is we have 30 poster presentations. And of course, with the Zoom situation, living in this COVID era, what we have done is we've divided these presentations into 3 different segments. So there are 10 poster presentations in basic translational and clinical sciences within this area, from everywhere from University of Miami, Florida International University, University of Florida, South Florida, etc. We have a second one on novel methods. And then the final one that we have is on therapeutics. And we have divided these, and what we did is we asked all the presenters for the poster presentation to share these posters on a box drive. This is a folder that we have shared with you. The 3 themes are presented over here. Within each of them, you'll find the poster itself, as well as an audio or a video that they have made to go along with it. And you'll find this information for all 3 of the sessions, the novel methods and therapeutics. And what we welcome you to do is to view these at any time in the next 30 days that you have the time to view it on your own, and you can reach out to the presenters as well. But today we wanted to take a opportunity to highlight 10 specific posters. And so we've invited these speakers, Steve Sato, Kevin Davis, Shreya Saxena, James Coy, Valentina Restrepo, Harrison Santiago, Annabelle Sierra, Federica Raciti, Andrew Cicirello, and Jerry Bonnell to sort of give us a very quick sort of a 3-minute pitch of their really exciting results from their posters. And so we'll just get started with that. So Steve, do you want to bring up your live presentation?
So basically, just to step through this, I think everyone is familiar with some of the challenges with implants and MRI scanning, and it's basically the interaction of the MRI fields with the implant, especially if the implant has conductive or magnetic components. There can be a very strong interaction, and as a result, we're currently limited to 1.5 Tesla in static magnetic field strength to prevent any damage to the probe and the patient. And back in 2012, I had a project in Brazil. We saw that our silicon carbide material at 2 Tesla in rats showed no evidence of image artifacts, whereas an MRI compatible probe, showed quite a bit of image artifact generation. So we had the idea that maybe we're onto something here, that this material would be relatively compatible with the MRI. Did some numerical simulations in ANSYS, looking at basically the absorption rate of energy into a phantom brain tissue. And we see that cubic silicon carbide, one of the forms of silicon carbide, performed extremely well below the FDA limit, whereas iridium, for example, or the noble metals, they tend to really heat up and cause damage to the brain. So the next thing we did was to study this further. Just had a PhD student, he's now in Switzerland and graduated in May, but we compared individual traces of heavily doped semi-metallic. You can dope semiconductors to be semi-metallic and they work pretty well as electrodes. These have a pretty low impedance, electrical impedance. And compared that to platinum, and we again see hotspots caused by the interaction of the MRI fields at 7 Tesla. So what's interesting about this is the magnetic susceptibility of the brain is pretty well matched to silicon carbide, also to silicon for that matter, whereas the noble metals have quite a large change in susceptibility, and this can cause image artifact stimulation. So we looked at artifacts based on the shift in this field component, magnetic field component at the probe, and we followed this up with actual experiments in a gel phantom in Moffitt and 7 Tesla, and we saw virtually transparent images from our silicon carbide material versus silicon, which showed pretty heavy image artifacts. That's kind of interesting. Doesn't mean that we're compatible, but it certainly is a very nice start to the story. So again, comparing these two guys here, we see our simulation results are tracking pretty well our experimental data. So more or less, we feel our code is giving good results. So then we took this 16-channel Michigan style all silicon carbide probe. We call it the all silicon carbide probe because on the business end that goes into the brain, there are no polymers and no metals. It's only silicon carbide as the support, as the conductor, and as the insulator. We only have metal on the packaging end, which would be outside of the brain. And looking at this at 7 Tesla, again, we see virtually no indication of where the probe even is. It looks extremely similar to the brain tissue on both coronal and sagittal cuts. So pretty nice result. So basically that's where we are. Our next step is to go in vivo in rodent. And we'd like to then measure any temperature rise, artifact generation, and of course do post-exposure and MRI and do some tissue histology. So that's where we are right now. I guess we're answering questions later, is that right? So I'll just stop sharing. And so thank you for everyone's attention. Hope it was interesting.
Thank you, Dr. Salo.
Thank you.
Our next speaker is Kevin Davis from University of Miami. Kevin, you can share your screen.
Okay, so I encourage you all to go look at the poster presentation. I'm not going to go over the whole thing. This was a ton of work by a ton of different people. So I'm going to focus on some of our recent highlights, the things that we did. And I'm working in Dr. Abhishek Prasad's laboratory where we investigate neural interfaces. And so I'll just touch on some of these highlights. So first, just some background. As mentioned in many of the talks earlier today, a brain-computer interface, it's a technology that aims to establish a direct link for transmitting information between the brain and an external device. Depicted here in this image, we have information coming from the brain can be collected and processed on a computer to actuate an external device or potentially environments used for electrophysiology and research studies for the brain and can be written back to the brain in the form of stimulation. So why is this helpful in our case for paralysis and specifically a patient with spinal cord injury? We know that spinal cord injury can affect more than 5 million people in the US. Individuals who attain it earlier in life can expect to have upward costs of $5 million over their lifetime. So this is a significant burden both on the individual, but as well as a society as a whole as a public health issue. So we're hopeful that brain-computer interfaces can aid patients with spinal cord injury to improve their independence and decrease the burden of disease. So now on to some of the points on why this is important, what we've done to combine this, use a BCI to help a patient with spinal cord injury. So here our big goal was to take it out of the laboratory, make it untethered. And as mentioned in the poster presentation that's associated with this was done after verifying that the system successfully worked in the lab. We then deployed this for at-home use by taking a mini computer and putting it into a 3D printed case, a mini computer and sticking it on the back of the patient's wheelchair. This setup minimized caretaker setup, one, because the neural sensing was fully implanted, and two, because we developed a mobile phone application that was associated with the system for the patient to use, and so that they could configure the system. And so all the caretaker had to do was put on a glove and turn on the computer to get it going. These components were all Bluetooth enabled for short range communication, not only taking this out of the laboratory, but they could also take it out of their home outside for use. And as seen in this video here, the data collection was all remote and it was patient initiated. So they were able to collect data on their own time. This eliminated the need for them to schedule transportation services to come into the lab and allowed us to collect more data on their time. And then also, while we have yet to systematically measure functional improvements, our patient actually sent us a video demonstrating that they were able to do things with the system at home that they were previously unable to do on their own. So we can see here that they're actually picking up a brush, adjusting the grip of the brush in their hand, and using it to brush their own hair without the aid of any assistance, which they hadn't previously been able to do. And so this work sort of highlights what we've been able to do to with a fully implanted brain-computer interface and deploy that at home with increasing ease of configuration and for practical use. So as there's improvements in neural sensing devices and wearable technology that will boost the utility of brain-computer interfaces, especially in the home. And this setup is also a methodology that sets up a fertile ground for long-term in vivo studies to answer potentially some really interesting engineering and neuroscience questions. So thank you.
Great, thank you, Kevin. Appreciate it. So with that, we'll move to Shreya Saxena from University of Florida.
That's great. So hi, everyone. Thank you for tuning in. First of all, an introduction, I'm Shreya Saxena, and I just started my lab here in the Department of Electrical and Computer Engineering at the University of Florida. I have a computational lab dealing with modeling high-dimensional neural activity and ongoing behavior using data analysis and machine learning methods. I also want to use these models to perform data-driven control, so that is to design stimulation strategies that can achieve a desired behavior. And today I'm excited to talk to you about one of my primary projects while I was a postdoc at Columbia University on the relationship between artificial neural networks and biological neural networks while we make movements at different speeds. So with that as a motivation, I want you to consider the case where you're practicing a new dance move, say. So you first practice it slowly, get the steps right, and then once you have learned it, you can actually flexibly speed up that movement and make it at the speed that you would like. There are many kinds of movements where this strategy applies. Today, what I want to do is address the question of how the motor cortex may be achieving this goal. So exactly. So for that question, a colleague, Abigail Russo at Columbia, trained 2 monkeys to cycle through a virtual environment while tracking a target. And this target, you can see on the screen, it was continuously moving at different speeds. And Abby also recorded single unit neural activity from the motor cortex and intramuscular EMG, so muscle signals. Okay, so what does the data actually look like? Here I'm showing the principal components of the muscle activity at different speeds, colored in different colors. And we can see that it looks fairly tangled up. So how do we actually begin to understand how this is generated? There's actually a growing trend to look towards neural networks when coming up with models of how the brain works. So we know that the motor cortex connects directly to the muscles. So to build a model of the motor cortex, we can directly train the network, an artificial neural network, to generate the output of the motor cortex. So this is exactly what we did. And this now provides a model for the motor cortex with the same inputs and outputs that the motor cortex would be getting. So let's see how this model does. So the trained model's activity, or the activity of the nodes, looks very different from the muscle activity, as you can see here. In fact, it has the signals that are necessary to generate the muscle activity given simple inputs. And analyzing this further and how, first of all, how is this model actually useful? So we can decode actually the neural signals, the motor cortex signals, better using the network activity than if we had just considered the EMG data. So here I'm showing an example neuron's firing rate and how that neuron's firing rate is actually how that neuron's firing rate is fit by the network activity as compared to if we had just used the EMG. And you can see that the network, the RNN here, recurrent neural network, fits that firing rate much better than the EMG. And this is not just true for one neuron. This is true for most of the neurons that we have. And so this is a population level effect. Moreover, so as you'll remember, this neural network didn't actually use any neural data while being trained, but we see that the population neural activity, so here is the population neural activity, actually looks remarkably like the model's activity. So without actually using the neural data, we can generate neural-like signals and basically models that are very useful for decoding neural signals. And this approach actually provides a testbed to eliminate the computational principles of the brain. This approach can be applied to cortex-wide data as well, which I'm excited about in the future. So hopefully I've convinced you that these kind of neural network models can be very useful. So this particular one can output EMG activity. It can fit single neuron activity and population level activity. However, I could have done that actually with a 10-layer deep neural networks, and that would not have helped us gain any insight on or in the understanding of the computational mechanisms involved in generating this activity. So the last thing we need from our model is actually interpretability. So why does it act the way it does? What characteristics of the model can we expect to be present in the data? And that's exactly what I dissect in the remainder of the poster. So here, I'm not going through details of that, but I basically dissect the neural network to find out why it acts the way it does and the dynamical principles due to which the same structure shows up in the neural data from the motor cortex. And this approach, again, is applicable in many different regions of the brain. Okay, so if you're interested in questions such as this, check out my lab, and of course, feel free to ask me any questions. I'm looking for collaborators and PhD students, and would just in general love to talk to any students who would like to learn more about approaches such as this, about this project or any other projects that I'm working on. Thank you so much for your time.
Thank you very much, Dr. Saxena. Our next speaker is James Choi from University of Miami. James, you can share your screen.
Yep, thanks. Hello, everyone. My name is James Choi. I'm a research associate with the Lee Lab at the Miami Project to Cure Paralysis. And today I'm going to talk to you about this work I've been doing titled Single Cell Analysis of the Cellular Heterogeneity and Interactions in the Injured Mouse Spinal Cord. And so as you may have already know, as shown in the schematic, after a spinal cord injury, axons and neurons are unable to regenerate through the injury site. This is due to a variety of factors, and one of those factors being the scar-like environment that forms. This scar-like environment is comprised of multiple cell types, such as peripherally derived immune cells, such as macrophages, colored in gray here, infiltrating fibroblasts, colored in green, and all this surrounded by a layer of reactive microglia and astrocytes. And so one of the ways that we can study the wound healing mechanisms in this process is to use RNA sequencing. In traditional RNA sequencing, what is done is you take tissue, you homogenize the tissue, you extract RNA, you map those sequences back to the genome, and then you can compare which genes are up or down regulated under different conditions. But what single cell RNA sequencing technology allows us to do is it allows us to see which cells are expressing which genes. So using this technology, we can extract RNA from each individual cell. We can see which genes are up or down regulated in each cell type under different conditions. And that really allows us much greater resolution in understanding wound healing. And the analogy that's commonly drawn is that bulk RNA sequencing is kind of like having the smoothie, whereas single cell RNA sequencing is like having the individual fruits that comprise the smoothie. And so there are a couple platforms for single cell RNA sequencing. One of the most common ones and the ones that we use in our study is a drop sequencing platform. Basically, under this platform, we dissociate the tissue enzymatically and mechanically to yield a single cell suspension. And this single cell suspension is then fed into a microfluidic device where each cell then becomes encapsulated in a lipid droplet. These droplets contain the reagents necessary for reverse transcription, as well as these single cell GEMs or gel emulsion beads. These beads contain a short oligonucleotide barcode, which is unique for each of these gems. And what that allows us to do is then track which mRNAs were captured from which individual cells. And so for this study, this is our experimental outline. what we did was we collected cells from various time points after spinal cord injury using a TA contusion model in mice. And the output of the sequencing platform are these matrices where we have data in this format where genes are along the rows, cells are along the columns, and basically we have information about which genes are expressed in which individual cells. And so this is kind of unruly to look at, and so what we do then is we organize this data using a variety of techniques borrowed from machine learning, such as dimensional reduction, clustering, and classification, in order to identify which cell types are present in our samples. And then we can perform a variety of differential gene expression tests to compare conditions, as well as look at which ligands are expressed by which cell types, which receptors are expressed by which cell types, and begin to infer some signaling pathways. And so this is the result of one of our study. This is called a UMAP. UMAP attempts to compress the information from those larger data sets into a two-dimensional figure. Here, every dot corresponds to a cell, and the color of the cell corresponds to the classification result, or basically the cell type that we've collected. And as you can see, we've collected a variety of cell types. And if you split the data by injury time point, you can see that this peripherally derived immune cell population, so monocytes and macrophages, is only really present in the injured spinal cord. But one of the strengths of single cell RNA sequencing is that we can further look at the heterogeneity within a cell type. And so, for example, what we've done in our study is that we've extracted all the vascular associated cells, such as fibroblasts, endothelial cells, and pericytes, and we re-ran them through the clustering pipeline to see what finer substates or subsets of cell types exist within this population. And with this analysis, what we found was that we were able to identify a rare population of cells called TIP cells. These cells have been known to be important for initiating angiogenesis. We see that their numbers dramatically increase after injury and subside or decrease over time. And based on our ligand receptor interaction analysis, we also find that these TIP cells are very important or very specifically expressing angiopoietin 2, which has been shown to be important for vascular destabilization. So that's a summary of my work. I want to acknowledge my lab, Dr. Pantelisoulas, for his comments, the oncogenomics core at UM, funding resources. I have posters at N8, and if you have any questions, please email me. Thank you.
Thank you, James, very much. So the next we'll have Valentina Restrepo from Biomedical Engineering.
Okay. Hello, everyone. My name is Valentina Restrepo. Our project is the development of a multi-channel galvanic skin response system to determine sympathetic level of injury and autonomic dysfunction. Our project deals with the diagnostic of the autonomic dysreflexia in a spinal cord injury patients. For lesions above T6, autonomic dysreflexia is common and it delays rehabilitation and impairs quality of life. Regardless of the site of the lesion, different levels of autonomic dysreflexia are present. Therefore, it is a need to detect the sympathetic level of injury. Currently, blood pressure over branchial artery is used in order to diagnose autonomic dysreflexia. However, it does not provide sympathetic level of injury. Other technique that can be used is galvanic skin response, known as GSR. GSR is a measurement of the skin conductance. This conductance is highly affected by the sweat produced from the eccrine sweat glands. The autonomic sympathetic system stimulates the sweat glands in response to any stimulus. Therefore, there is a need of a safe and reliable GSR multi-channel system. A schematic of the system that we develop is shown on your right. First, we have a 22 millivolt 75 hertz sine wave generator. The voltage is applied to the four common electrodes corresponding to the 4 channels. These electrodes are placed on different sides along the side of the spinal cord. The current will flow through the body to a second electrode, which is connected to a transimpedance amplifier that converts current to voltage. Using this architecture, we achieve a safe and site-selective GSR multi-channel system. We perform bench and pilot testing on subjects. Figure 1 shows a long typical GSR expected response, and Figure 2 shows a short data acquired with our system on a subject. We enlist the GSR responses using noxious auditory stimuli, and to assess selectivity, we verified that one channel response was reproducible regardless the location of the other channel sites. In conclusion, we have developed and tested a multi-channel GCR system, which is a safe and selective to be used in patients that suffer from autonomic dysreflexia. We have an approved IRB to record spinal cord injury patients. To record spinal cord injury participants with 4 simultaneous channels. Thank you.
Thank you, Valentina. So our next speaker is Harrison Santiago from University of Florida. Harrison, you can go ahead.
So, hello everyone. My name is Harrison Santiago, and I'm an undergraduate research assistant in the neuroprosthetic lab under the direction of Dr. Kevin Otto. So as a bit of a background, type 1 diabetes is an autoimmune attack that affects 1.25 million Americans and millions more worldwide. It works by causing the loss of beta cells in the pancreas, which are necessary to maintain glucose homeostasis. However, some beta cells remain, and they have been found to secrete insulin in response to acetylcholine, a neurotransmitter released in response to vagus nerve stimulation. And although there is already literature exploring vagus nerve stimulation as treatment for type 1 diabetes, they explored cervical or subdiaphragmatic stimulation, while we are looking specifically at pancreatic neuromodulation. So in short, we analyzed the effect that the amplitude of the vagus nerve stimulation has on blood glucose levels in a diabetic environment. So to gather data, electrodes were implanted on the pancreatic branch of the vagus nerve in Lewis rats, and telemeters were implanted to measure blood glucose. A stimulus was then applied using various amplitudes, and changes in blood glucose were measured. In individual trials, there was a high amount of variance between the blood glucose trends, and even looking at the aggregate of the entire data set, we see that even divided among different amplitudes, there is still a decent amount of variance and nonlinearity. So these characteristics make it difficult to analyze using traditional techniques. So to gain some insight, I applied a multiple regression machine learning algorithm and relative weight analysis. The idea behind multiple regression is that you have multiple predictive features that you use to calculate an output. And the goal is to create a function that minimizes the difference between the output and whatever variable you are trying to predict. And a good resulting function acts similarly to whatever real-world mechanism is generating that dataset. Once you have your optimal function, it provides weights or a numeric value which is associated with the importance of each predictive feature. So in our case, some of the predictive features were the changing amplitude and the initial blood glucose levels. And after optimizing the function, the weights indicated that the amplitude could have a substantial impact on blood glucose levels. From here, we turn to relative weight analysis to provide a confidence interval showing that the weights are statistically significant. And this process works by creating a new predictive feature that is uncorrelated to the dataset. The idea is that this should have 0 importance, and you can then use that as a baseline to judge your other features off of. And after doing this, we could generate a confidence interval that supports that the amplitude of vagus nerve stimulation does have a significant impact in lowering blood glucose levels in a diabetic environment. As a bonus, we could also use the regression function to predict blood glucose levels 15 minutes out with an average error of 3.3% and 30 minutes out with an average error of 4.7%. So if you have any questions about our data analysis, I'll look at the poster, or I'm happy to take any questions in the Q&A session. Thank you.
Thank you, Harrison. Thank you for joining us today. So next speaker, the poster presenter will be Annabelle Alvarez-Sierra from Abhishek Prasad's lab.
So my name is Annabelle Alvarez. I work at the Neurointerfaces lab for Abhishek Prasad. And today we're going to be talking about oxidative stress in intracortical implants. As Kevin said before, brain control assistive technologies use neurointerfaces to record neuronal signals over long periods of time, and this signal is not stable and it degrades over time due to biological and non-biological factors. Microelectrodes as UTA arrays are invasive and produce a strong foreign body response since they are inserted directly in the brain. And oxidative stress is part of this response. Understanding how the tissue reacts to this type of microelectrodes is important for identifying different routes for future therapeutics approach. In our project, in our lab, we basically try to study and understand what's happening at the tissue level. In this project, we wanted to look at the expression of genes related to the NADPH oxidase complex activation, which is a producer of reactive oxygen species in the tissue. And we use rats implanted with UTA arrays, and we look at the gene expression using PCR from tissue extracted at different acute time points up to 7 days. In general, like what we saw on our results is that gene expression of membrane and cytosolic components of the complex remain significantly upregulated across all these time points. We also look, we have NOX2 and NOX4 and NCF1, which comprise the NADPH complex. And we also look at nitric oxide synthetase, which produce nitric oxide, and the production of this creates also reactive nitrogen species that in combination with reactive oxygen species create a more oxidative environment in the tissue. On the other hand, we wanted to look at what's happening to the antioxidants production, and our gene expression for different antioxidants show that, on the contrary, they're not being expressed as much. The values remain on their baseline values and for most of the time points. So there's like an under-regulation for antioxidants in comparison for the proxyans production. So from this, we basically concluded that our results suggest that from the gene expression, NADPH complex is being activated in the tissue, and it may be producing great amounts of reactive oxygen species that at the same time, because we have an imbalance on antioxidant production, so the redox, like the redox balance is not maintained, these reactive oxygen species are accumulating in the tissue. And what may be happening is that the accumulation of the reactive oxygen species is causing oxidative stress that is damaging the cells and is making the tissues surrounding the electrode not viable to last long time recordings. So this is like the overall of our study. We had a poster like that has been shown on the folders. If you guys have any other questions, I'll feel free to ask, and I can answer any question that you have. Thank you for listening.
Thank you, Annabelle. Our next speaker is Federica Raciti from University of Miami.
Okay, so hi, my name is Federica Raciti. I'm a PhD student from Dr. Rajguru's lab. And today I'm going to give you just a brief overview about my poster. You can find it in the box folder, which is basic clinical and translational on the cloud. So in our lab, we use IR stimulation as a tool to modulate the activity of the vestibular system. And in particular, my work focuses on the study of the cellular and molecular characteristic underlying the mechanism of response to IR stimulation in the vestibular neuroepithelium. In this work specifically, what we do is that we use the well-known regional morphological characteristic of the utricular macula combined with the study of the variety of response to infrared stimulation in order to determine, to gain further knowledge about the spatiotemporal selectivity of this novel tool that is IR. So here, okay. So here there is the experimental setup that we use. We use female Longevans rats, and we access the inner ear surgically. We then identify the utricular macula and target and insert a 3 optical fiber bundle targeting the utricular macula itself. Then we deliver the IR radiation through each one of the fiber alternatively and record the corresponding eye movement evoked. We then analyze different parameters from the eye movement evoked. We look at magnitude and latency, and from the results that we have obtained so far, only magnitude seems to be dependent by the localization of the stimulation site. So in 80% of the animal present in our experimental group, a stimulation occurring towards the caudal region of the utricular macula actually seems to cause a larger response compared to a stimulation occurring in other regions of the vestibular neuroepithelium. To further analyze the stimulation site, what we did is that we caused a lesion on the tissue in corresponding to the region that we were stimulating at a time, using an ablation along the trajectory of the beam path that was giving us the largest response. We then harvested the sample, stained, and imaged at the confocal. So here is an example of the samples that we imaged from rat 1, which is the same rat from the data that I was showing you before. And this is, okay, this dark area is the lesion site. So we confirm that the largest response was given by a stimulation occurring towards the caudal region. But also what we saw in our staining is that the stimulation occurred towards an area, which is the striatal area highlighted here by this white line, which is a region of the utricular macula that is known to be enriched in type 1 hair cells, suggesting that type 1 vestibular hair cells might be the primary driver of the photothermal response. And this also confirmed our previous hypothesis. So overall, the main take-home message of this study is that the diverse range of IR photosensitivity shown by the hair cell type within the vestibular neuroepithelium can make focal or multi-point IR light stimulation a valid tool that can be used by researchers to delineate functional differences between the hair cell subtypes and regions in the vestibular end organ. So with that, I'd like to thank my lab, my mentor, and the people, all the people in my lab, our collaborators, and the funding agency, and the Institute of Neural Engineering for giving me the possibility to talk about my work. So if you have any question, I sadly won't be able to attend the Q&A session, but you can find this presentation on Box, and there is my email address, and you can email me there. Thank you so much.
Thank you, Federica. All right, so we have Andrew Cicerello next.
All right, thanks, Dr. Rajguru. Should be in full screen now. Yeah, so as Dr. Rajguru said, I'm a graduate student in the Department of Biomedical Engineering, and I'm really excited to be detailing our lab's development of a synthetic ligand receptor binding system for targeted delivery to the spinal cord. So as many of you are acutely aware, spinal cord injury is an incredibly complicated tissue engineering problem with no current widely available treatment option. And so in tissue engineering, one viable option that a lot of people have been discovering is, or investigating, is targeted drug delivery. And this has proven to be successful in a number of injury dynamics, but is still relatively complicated in spinal cord injury. And that's because a lot of the targeted drug delivery systems take advantage of naturally occurring targets. And so this is problematic in the spinal cord as they are either widespread targets throughout the whole central nervous system, and the tissue is susceptible to remodeling over time, altogether creating a limited window for therapeutic delivery to the injured spinal cord. And so our lab tried to think about this in a different way and develop a synthetic system that could hopefully ameliorate some of these design challenges that are present with natural targeting systems. And so to do that, we developed a two-part system. where the first part is our receptor binding depot. And this is modified from our PEG hydrogel tubes that act as an axonal guidance system. And they're modified to express a binding receptor throughout the length of the tubes. And this would be implanted immediately after a spinal cord injury. And we showed fine control over the concentration of the receptor, which then goes into our second part, which would be our ligand functionalized nanoparticles. And these functionalized nanoparticles have the corresponding ligand that would bind to the receptor implanted at the injury site. And they're fabricated from PLGA PEG and fabricated to have a diameter of approximately 150 nanometers. And so to test our system, we started first in vitro, and we incubated our binding depot with our nanoparticles, and we showed that not only did the nanoparticles bind to our binding depot assessed by fluorescence on the ligand, they did so in a dose-dependent fashion, meaning we could deliver multiple rounds of therapeutics or multiple rounds of nanoparticles. And so from there, we moved into an in vivo model where we performed a mid-thoracic hemisection in a mouse and implanted our binding depot immediately after injury. And 3 days later, intravenously injected our nanoparticles. And we saw that in our conditions that got the receptor implant preferential binding at the spinal cord compared to the implant with no receptor and sham-only control conditions. And this was also reflected in the histology, where on the left we have our injured tissue, on the right we have our healthy tissue, and we can see FITC conjugated nanoparticles present in our injured region. And so moving forward now, we're really interested in investigating the biodistribution properties of our nanoparticles and potentially delivering a therapeutic factor in the future. And so with that, I'd just like to thank my advisor, Dr. Courtney Dumont, our funding sources and collaborators, and I'd be happy to take any questions during the question and answer period. Thank you.
Thank you, Andrew. So we can conclude this live poster presentation session with Jerry Bonnell from University of Miami.
So hello, everyone. Thank you so much for inviting us to the Neurotrauma Symposium this year, as well as to the live poster presentation. So my name is Jerry Bonnell. I'm a PhD student from the Department of Computer Science at the University of Miami, and I'll be presenting our work on developing a stacking model for the classification of multiple therapies following experimental brain injury. So traumatic brain injuries, or TBIs, are a major cause of death and disability. And the heterogeneity of the TBI population remains a challenge for identifying effective treatment strategies. And so in our work, we evaluate whether machine learning, which is capable of overcoming limitations in univariate analyses, can help to perhaps overcome this barrier. And so in our previous work, we demonstrated that supervised pairwise classifiers can correctly discriminate between therapies the animal received when given recovery metrics recorded post-experimental brain injury. And so we build on in this work, we build on the strength of these pairwise classifiers by assembling them together into a stacking scheme to correctly classify multiple treatment groups. And so the proposed architecture consists of 2 layers. So number 1, we have a number of multi-layer perceptrons, or MLP networks, as individual estimators, fitted on pairwise combinations of the classes from the training set. And at our second layer, we have a support vector machine, or SVM, as a final estimator, which is fitted on a redescribed data set, which is composed from the output from these individual estimators. And some may notice that since the first layer is pairwise, the stud does rise a possibility for a large number of contributing features to the master classifier. And so in order to control for this, we apply principal component analysis, or PCA, to help overcome this problem. And so the results of our model are quite promising when evaluated on 2 different data sets, one with 3 treatment groups and another with 5 treatment groups. And most pointedly, we observed that as we move from the 3 treatment group data set to the 5 treatment group data set, the degrade in performance is much more gradual, with a reliable 12% improvement. And another finding for us that which we're quite excited about is that when we visualize the decision boundary at the master layer with TSNE, which is a dimensionality reduction tool for visualizing high dimensional data, for both the 3 treatment group data set as well as the 5, we observe visible clusters which are separated by treatment group for both of the data sets. So this is exciting work, which is quite consequential for advancing more algorithms that can help, again, address this difficult problem of addressing the heterogeneity, which has been a huge hindrance to developing or for discovering any kind of cures or for the TBI population. And so with that, yes, I would like to thank you all once again. Unfortunately, I will not be able to attend the Q&A, but our collaborator, Hannah Radabal, will be there. So I'll point you in her direction. And of course, my email is also unboxed. So if you have any questions, feel free, and I'd be happy to address them. Thank you again.
Appreciate Jerry, and thank you again to all the poster presentations, poster presenters, including the one that we were able to highlight here. It would have been great if we could have sort of met in person, and all of us could have sort of been in a same room and sort of go through the posters like we are used to doing in a Society for Neuroscience meeting, for example, or even the previous versions of the Neural Engineering Research Symposium. But really exciting science, exciting work that's being done. What I wanted to just take the opportunity to do is to say that, in addition to the live poster presentations that we did, we had a committee made up of graduate students and assistant scientists, the organizers, the three organizers who have led the entire symposium, evaluate all of the presentations, all the posters for 4 categories. So it was for innovation, significance, the presentation, as well as translational potential for the research. And we have some poster awards to announce from the scores that have been received. So from the Institute for Neural Engineering, I want to congratulate Beatrice from Florida International University, Melissa from University of Miami, and Ladan Jurasek from University of Florida for receiving the highest scores in all of the categories. And so we'll reach out to you and we'll be sending you a certificate as well as a $250 gift card, sort of as a congratulations for your poster awards. And as well, the Clinical Translational Science Institute at the University of Miami, they judged the posters for the translational potential and has decided to award 3 posters, Kevin Davis, Jerry Bonnell, and Samantha Rincan Sabatino from the University of Miami for their awards. And so all 3 of you will also be receiving a certificate as well as a $250 gift card from the CTSI at the University of Miami. I really want to thank the CTSI, but also congratulations to all 6 poster awardees for the excellent job putting the posters together. And of course, all of you, thank you very, very much again for putting your posters online. With that, what I want to say is that we're sort of nearing towards conclusion. If you have any questions, any thoughts for any of the speakers, any of the poster presenters, they're all here. And so you can either use the Q&A function and post your questions, or you can go on the box folder, view their presentations. And Erica just shared the box folder with all of you in the chat. You can go there, you can view them at your pleasure. This link will be open for the next 30 days. And all of you will have access to it to view all of the videos and all of the posters. All of the speakers' emails are listed over there as well. And so you can go view those and reach out to them via email. Again, as I said, unfortunately, given the situation, we couldn't do everything in person, and we would have loved to do that in person. So there's much more interaction. But we're trying to do the best we can and adapt in this online world. So if there are no more questions, we'll keep the Zoom open for the next 1 hour. But I want to thank Dr. Dietrich for supporting the Institute and leading it. I want to thank Dr. Rachele Sangaletti, Shu Pan, and Hannah Radbo for really all your hard work, along with Federica Rocchiti, in setting up the entire symposium, reaching out to the speakers, organizing it. I want to thank Ignacio and Erica for really helping with all the logistics for the conference. Without you both, we couldn't have done all of this. And I want to thank the University of Miami, the CTSI at the University of Miami, and of course, all the wonderful speakers from University of Florida, Florida State University, Florida International University, University of South Florida, as well as Max Planck and FAU, who joined in and gave wonderful, wonderful presentations. Thank you again very, very much. And with that, I'll sign off.
Thank you all. Incredible meeting. Just went too fast, but next year we'll have some new ideas to move the research even further forward. Thank you all.
Yes, if anybody has any suggestions, any thoughts on how we can do this, hopefully next time in person, and new topics that we can bring in and new speakers that we can bring in from our institutions, please let us know.